Hello everyone, welcome to today's tutorial. In this video I am going to show you how to perform docking using Autodock Vina. Before we start we need to install required software and tools. In this page you can see what are the things needs to be installed. The first one is Autodock Vina and followed by Mgltools, BIOVIA Discovery Studio and Open Babel. Also I have provided the overall flowchart for the understanding. The first step is protein preparation. The next step is ligand preparation. Then we need to do docking using Autodock Vina. For visualization, we are going to use BioVia Discovery Studio Visualizer. So now let's start. Go to the tutorial. Okay, for docking, first we need a protein structure. We can download the protein structure from protein data bank. You can just simply type PDP, which means protein data bank. Once it's open, go to the PDP homepage. Okay, in this search panel, you can search whatever protein you're going to work on. So here I'm going to type SARS-CoV-2. So it came here SARS-CoV-2 virus. So just click and I'm going to take any one of the structures from here. And okay, I'm going to take this structure, a dual inhibitor against main protease. So the PDP ID is 7V1T. So just click it. Here you can check the experimental data types. So the method is X-ray diffraction and the resolution is 2.56 Armstrong. So if you want to know more, you just scroll down. So here you can see the macromolecule informations like the molecule name, the chain names, and the sequence length, and the organisms, and the details, and also the images. So just scroll down. If you can see the small molecule informations also. So there are one ligand is there in this macromolecule structure. So once this paper get published, they will provide the article informations also. So just go to the download files and click. So we need to download this in PDP file format. So just click PDP format. Okay, the protein structure has been downloaded. So now we need to download our ligand molecule. So for that I am going to use PubChem database. Just type PubChem. So just click the home page. In this search engine we can search whatever chemical structure we want. So I am going to use curcumin structure. So just type curcumin. Okay, here you can see the best match of my keyword curcumin. So this is the best match. So I'm just click the first one. In the right side corner we have the content. Here they will provide all the details about the curcumin compound. So if you want you can search all the details of the particular compound. Okay, once you read about the contents, just click download. So here they will provide so many options for downloading the chemical structure. We need only the 2D structure, 2D structure in SDF file format. So just click SDF. So my chemical structure has been downloaded. So once we download the protein structure and the chemical structure, we need to make a new file folder for working on the docking. So in desktop, I am create a new folder called doc. So I'm going to move my protein structure and ligand structure to here. So I'll just cut these two files and keep it in my working directory. So this is my protein structure 7B1T and this is my Ligand compound. So I am going to rename this Ligand compound as curcumin. So the protein structure is in PDB file format and the curcumin structure is in STF file format. So we for docking using Autodock Vina we need a pdbqt file format. So we need to convert the PDB file format into pdbqt file format. For this we are going to use MGL tool for converting the PDB file into pdbqt file. And for the ligand, before we start, we need to convert the STF into PDB file format. For that, we can use OpenBabel. OpenBabel also free source. You can use it in online. Just search OpenBabel. Just scroll down. Or else, if you want, you can download the OpenBabel software. Then you can use your command prompt to convert the STF file format to PDB file format or pdbqt file format, whatever file format you want to. I am going to use the command prompt. So if you did not install the OpenBabel, you can convert it here. I'm also provide the hyperlink in the tutorial itself. Okay, so now I'm going to convert the STF file format into PDB file format using command prompt because I already installed the OpenBabel. So just I'm going to type cmd. Command prompt will be open. Then I'm going to copy this location. Copy it. Then I'm going to change the directory here. cd and I paste the file location. So now I am in my working directory. So if you want to check whether the OpenBabel install or not, just type O Babel, not OpenBabel, just O Babel and enter. Okay, it's installed and the current version is 2.4.1. So once it's done, just check your file. Type dir. It will show all the files in your working directory. So this is my ligand. It's in STF file format. I'm going to convert this curcumin.stf file format to PDB file format. For that, just type O Babel input curcumin.stf and I need a output of curcumin.pdb. Okay, in output the O should be capital letter and I'm going to add hydrogen. So hyphen H must be there. Then enter. It shows one molecule converted. So just minimize the command prompt. Check whether it is converting or not. Yes, it converted into PDB file format. So now we have protein structure as PDB file format and ligand structure as PDB file format. Now we are going to convert this PDB file format into pdbqt file format. Okay, for that you have to open the Autodock tools. Once you install your MGL tool in your desktop, you can see Autodock tools like this. You have to open this Autodock tool. It's opening. Okay, once it's open, the first thing you have to do is set your working directory location. For that, go to File, Preferences, Set. So remove the existing startup directory. Go to your working directory, copy the location, just click it here. It will show the location. Then copy this location. Go again, just paste it here. Then click Set, Make Default. So now whatever files we are going to read, it will directly take from our working directory. So go to File, click Read Molecule. First we are going to read our protein structure for preprocessing. Just click Read Molecule. So I am going to take this one, 7B1T. Click Open. Okay, this is my protein structure. I am going to preprocess this structure first. First we need to delete the water. For that, go to Edit, Delete Water. So sometimes we no need to delete the water molecules. It depends on your protein function. So not always delete water will work. So read the literature which they provided in your. Okay, sometimes we don't need to delete the water because sometimes the water molecule play a major role in the active sites. So that time you need to keep the water molecule in your active sites. So once you deleted here, I don't need the water molecule, so I have deleted. Once you deleted the water molecule, the next step is we need to add the hydrogens. For that, click Edit, Hydrogens, click Add. Here, click Polar only, then click OK. And the next step is we need to add the charges. Click Edit, go to Charges, add Coleman charges. And it shows the total Coleman charge added is equal to 3.061. Okay, click OK. Again go to Edit. We need to add the atoms, like assign ready. First you click the assign ready, then you have to select the assign AD4 type. AD4 type means Autodock 4 type. First I am adding the assign ready. Click United ready. Again go to Edit, Atoms, assign AD4 type. Okay, now this molecule is ready to converting into pdbqt file format. Go to File, click Save, write pdbqt. Here you just click Sort Nodes, then click OK. Okay, the PDB, now we pre-process our protein structure for docking. Also, we converted the PDB into pdbqt file. So if you want, you can check your working directory. Here, the 7v1t, it's in pdbqt file format. Okay, now we need to, after pre-processing the protein structure, we need to make a grid box over the protein. If you know your active sites, click the plus sign, click the H sign, and select the amino acid regions of your active sites. So it will make yellow color dots on the protein, whatever place you are selecting. Here you can see this the amino acid regions I have selected here. Okay, so if you know your active site amino acid, just click it here, then make a grid box only to this particular sites. So here I am going to make a grid box for the entire protein because I am going to do a blind docking. So I just unclick all of them. Just click grid, macromolecule, then choose. I'm going to select this protein, then select macromolecule. So it shows contains no non-bonded atoms. Click OK. It will ask you to save it again. So just _1. Okay, then again go to grid box. Okay, I'm going to cover the entire protein. The protein pre-process has been done. Now we are going to prepare our ligand molecule. So we need to delete this protein file. Go to edit, then click delete all molecules. Click continue. Okay, now we need to import our ligand molecule. We have converted our ligand molecule to PDP file format using Open Babel. You just import the output file here. Go to File, Read Molecule, import the curcumin PDP file format. Okay, it's open. Okay, this is our PDP file format of the curcumin. Then click Ligand, click Input and choose. Click curcumin. Select molecule for Autodock 4. So it shows setup curcumin added gastric charges, detected 12 rotatable bonds. Then click OK. Then again click Ligand, click torsion tree, choose root. Again go to Ligand, torsion tree, detect root. Okay, it's detected the root. So now we need to save this file as pdbqt file format. Go to Ligand, click output, save as pdbqt. Okay, then click save. Autodock Vina need a config file with all the details of our ligand name and the receptor name. Also, they already made one. Also, I have provided the file in the description. So just open the config file and also the grid dimension file. So in the config file, you have to specify the receptor file name and the ligand file name. And the center XYZ and the size XYZ, you can get it from grid file. So come to the config file. So my receptor name is 7v1t dot pdbqt 7v1t. So I just copy the file name and paste it here. Also my ligand name is curcumin.pdbqt. The center XYZ, open the grid, go to the center, just copy all the coordinates and paste it here. X, Y and Z. For the size X, go to the grid file again, copy the NPTS. Just copy and paste it here. Size X, size Y and size Z. And the energy range should be 4 and exhaustiveness should be 8. Make it default and save the file. So now we all set to performing the Autodock Vina. So before that we need two files. Once you install the Autodock Vina, go to program file, just find the Scripps Research Institute. Here open Vina, Just copy these two files, Vina and Vina split. Copy this file and paste in your working directory. I pasted here. Now we need to check whether the Vina is working or not. Just copy the file location, open the command prompt and change directory, paste the file location. Just type vina.exe, vina.executive, so vina.exe. So if you are getting usage like this, it's perfectly working in your system. So the next, to check all the files in our working directory, type dir. It will show all the files. Now we need to give one command for performing the Autodock Vina. Just vina.exe hyphen hyphen config, then our file name config.txt. If you save your config file in some other name, use the name then .text, and we need a output file for this docking. So just hyphen hyphen out paste curcumin underscore doc pdp. I am choosing pdp. If you want, you can use pdbqt also. Either way it will work. Then hyphen hyphen. We need one log file to check the docking scores. So for that, log.text. Now just press the enter. It will start running. Okay, it shows detector 8 CPU, reading input and setting up the scoring function. It's all done now. The binding site is analyzing. So once it's done, it will show the docking score. Then we are going to visualize the top compound using Discovery Studio. Wait for some time, it will take time, 2 or 3 minutes. Almost done. Alright, it's done. We got the docking score of minus 5.9 and it generates 9 models. Now we are going to open the first model in Discovery Studio. Before that, we just check what are the files it generated. Just minimize it. Just okay, this is our working directory here. See, here you can see the log file. Just open the log file. So here you can get the docking score of the all models. So the first model docking score is minus 5.9 and the last model docking score is minus 5.0, which means it will go through more conformation states. Then it will try to bind the active sites and produce the docking score. So I just closed it. Okay, after this, just open the Discovery Studio visualizer. Go to File, Open, go to your working directory. Okay, first open the pdbqt file of the protein structure. Okay, click all files. Here the pdbqt structure of our protein. Click open. Okay, then again go to file, click open. So this is our results file, curcumin underscore doc. Click open. Then click Control plus H. Okay, here it will show all the model of the docked compound. I am going to copy the first model only. So click the first model, then copy the file and again Control plus H, paste it here. Okay, I have done the blind docking, so my compound has bind this particular site. So if you have an active site, it will go and bind some other place. It depends on your protein structure and the active site. So again click the curcumin dock model 0. Then here define ligand. Okay, once you define the ligand, click the ligand interactions. Okay, here it will source the ligand interactions. Okay, if you want to see the 2D diagram of the interactions, just go here and click show 2D diagram. Okay, this is the amino acid interactions between the curcumin and the SARS-CoV-2 protein structure. So once it's done, you can download this particular image also, or else you can copy and paste in Paint.